In clip 3, we will continue with another set of core hallmarks, and these hallmarks facilitate dissemination or spread of cancer cells. The hallmarks that we will discuss are inducing angiogenesis and activation, invasion and metastasis. Let's start with angiogenesis first. As you may understand, cells and also cancer cells need nutrients and oxygen. So there is an elegant mechanism called angiogenesis that allows the formation of blood vessels. Angiogenesis is the formation of new blood vessels, and these new blood vessels can assure a supply of nutrients and oxygen to the cancer cells. Therefore, angiogenesis can support neoplastic growth, and in most of the cases, angiogenesis occurs quite early in the process of cancer development, because without these newly formed blood vessels, the cancer cells do not have any nutrients and oxygens, and therefore the cancer cannot fully develop. Angiogenesis is also considered an important target for therapy, and I will show you some examples later on. First, I would like to discuss again an important factor, and that is vascular endothelial growth factor, or VEGF in short. VEGF is the growth factor that can induce angiogenesis. As a consequence of hypoxia, that is a lack of oxygen, or other factors secreted by the cancer cell, VEGF will be expressed and promote the development of new blood vessels. The formation of these new blood vessels is not only important for cancer growth, but may also play a role in cancer spread or metastasis, because cancer cells that can enter the bloodstream may travel to other sites of the body and form the metastasis over there. Here I would like to show you some examples of scientific research and clinical diagnostics in the field of angiogenesis. What we see on the left is a dorsal skin fold chamber that is used to study the angiogenic process. In this animal model, the dorsal skin fold chamber is used as a non-invasive method. Only the skin fold is put in the middle of the small window. There is no surgery or another procedure needed, and the blood vessel formation can be easily followed by eye or under the microscope. If the animal would receive specific drugs or maybe even specific dietary interventions, it would be possible to study the effects on angiogenesis. Here you do see a microslide, and this is a small slide containing culture medium that can be used to culture, for example, endothelial cells. Endothelial cells are the cells that form the wall of the blood vessel, and so you can study the effects of drugs or nutrients on the formation of these new blood vessels. And finally, what you see on this slide is an example that angiogenesis can also be used in the clinic, for example, as a measure for treatment efficacy. What we see over here is an MRI scan of a patient with breast cancer. The left breast is affected, and we clearly see that the blood flow and the activity in the tumor can be visualized. On the left, in panel A, we see the patient before the treatment started, and on the right, we see the same patient, but now after 4 weeks of treatment with an antibody against VEGF. So the angiogenesis is inhibited, and that is clearly seen on this figure. We will now continue to the next core hallmark, which is activating, invasion and metastasis. Activating, invasion and metastasis is actually the spread of cancer cells. I already explained you that spread of cancer cells can be facilitated via newly formed blood vessels, but also via lymphatic vessels. The cancer cells can spread to distant organs, so other places in the body, and this is again a multi-step process. It is important to realize that each tumor has its own preferences and latency time, meaning that each tumor has a preference for a site of a metastasis, and it also depends on the tumor and of course the aggressiveness, how long it takes before a metastasis can be formed and detected. So what you see over here are a few examples of primary tumors, so the first tumor in the body, and then the preferred site of metastasis. So for example, for patients with prostate cancer, we know that if they develop a metastasis, this often occurs in the bone. For breast cancer patients, metastases are mostly found in lung, liver, or brain, and the same is true for melanoma, which is a form of skin cancer, colorectal cancer, large bowel cancer. If there is a metastasis, it can be mostly detected in the liver and lung, and so on. The process underlying the development of a metastasis is called the invasion metastasis cascade. It all starts, of course, with the tumor at the primary location. One of the definitions of a malignant tumor is that it starts invading surrounding or neighboring tissue structures. So now we talk about local invasion. At a certain moment, it might, however, be possible that cancer cells also migrate towards the blood vessels and even invade into the blood vessel. This process is called intravasation. The cancer cells in the circulation can travel through the body and at certain places leave the circulation again. This is called extravasation. At this new location, the cancer cells can start growing again, forming a metastasis, first small, a micrometastasis, but eventually becoming a new tumor, which is called macrometastasis, and which can be clinically detected. If we zoom in on this invasion metastasis cascade again, we do see that there is an important molecular process that facilitates the metastatic behavior of the cancer cells. This process is called EMT, or epithelial mesenchymal transition, and I will explain in the next slide what it means. I actually would like to emphasize one more time the importance of the invasion metastasis cascade, meaning that there is local invasion, intravasation, extravasation, micrometastasis, and macrometastasis. The epithelial mesenchymal transition is actually a physiological process in which the phenotype of the cell changes. First, it should be said that many cancers, and especially the common ones such as breast cancer, colorectal cancer, and prostate cancer, usually arise from epithelial cells. Therefore, they are called carcinomas. These epithelial cells have the characteristics that they are strictly organized. There is a fixed border, neatly outlined, and cells are attached to each other by cell adhesion molecules. During the epithelial mesenchymal transition, these properties might be lost because the cells start to look like a mesenchymal cell. A mesenchymal cell is not well organized. They can travel and migrate. And this is actually quite useful during embryogenesis, when new organs needs to be formed, or during wound healing, when tissue needs to be replaced. However, in the case of cancer, epithelial to mesenchymal transition might also contribute to the metastatic process, because you can imagine that these mesenchymal cells can travel. The reason that the phenotype is changing is because specific markers are lost. Ecadherin is one of the cell adhesion molecules that is highly present in epithelial cells and is responsible for these cells holding each other. During the epithelial mesenchymal transition, the expression of this marker is lower, meaning that the marker disappears. On the other hand, other markers that are specific for mesenchymal cells, such as vimentin, appears on these new cell types. So what we see in this figure is a visualization of the EMT, or epithelial to mesenchymal transition process. We start over here with the malignant or pre-malignant cells, which are epithelial cells. Once their phenotype, their characteristic changes into a phenotype that is more like mesenchymal cell types, the cells acquire the capability to migrate. They migrate, for example, into the blood or the lymphatic vessel. So this is again the process that allows intravasation into the circulation. Once the tumor cells are now in the circulation, they can start entering new sites, so new parts of the body. We do have this mesenchymal-like cell over here, And once the mesenchymal cell is entering the new tissue, the site of the metastasis, the inverse process can actually occur, and this is called MET, or mesenchymal epithelial transition. So now the mesenchymal cell becomes like an epithelial cell again, starts growing and forms a new tumor. The effects of epithelial to mesenchymal transition, as well as the invasion metastasis cascade, are also clearly visible in these pictures. So what we see on the left is a bone scan of a patient with prostate cancer. And as you might remember, prostate cancer tends to form a metastasis in the bone, which is also clearly visualized as these dark dots shown over here, and all these other spots. The second picture shows a liver of a patient with a colorectal cancer, but the colorectal cancer has now also spread to the liver, which can be seen as these areas which form the metastasis in the liver of this patient. So what we have discussed in this clip is the question, how do cancer cells spread? And in order to be able to understand that, it is important to recognize the importance of the invasion metastasis cascade, as well as the molecular process underlying this cascade, and that is the epithelial mesenchymal transition. The role of this EMT process in the development of metastasis is important, and the same is true for the inverse process, in which the mesenchymal cells become like epithelial cells again.